Good afternoon, everyone. I'm MIRNA, this is Matien, and we're here to reinvent early-stage cancer diagnosis. Cancer is one of the deadliest diseases globally. 1 in 3 of us sitting here in this room are likely to get cancer at some point in our lifetime. But here's the thing, many types of cancers are actually curable. Statistics clearly show that recovery rates and treatment efficacies are more than 3 times when cancer is diagnosed early. So to address this, liquid biopsy-based technologies were developed. Liquid biopsy basically involves taking either the blood samples, urine, or sputum, and picking out either cancer cells or novel biomaterial like DNA or microRNA. So very recently, a novel class of genetic biomaterial called the microRNA was discovered. We like to think of microRNA as the devilish little cousin of DNA, structurally similar, but a lot smaller, and so a lot harder to detect. To date, 2,500 different types of microRNAs have been discovered and found to have a strong correlation with several different types of cancers. Excited by these prospects, biotech companies started jumping on the bandwagon and developing microRNA-based kits to do their detection. But this was met with some problems. The technology that they used was PCR, which was originally meant for the detection of longer strands of DNA. So to suit this to smaller microRNA, it meant increasing the number of steps, thereby complicating the workflow. And as I mentioned, microRNA was a lot smaller. So while PCR could reliably distinguish different types of DNA, it could not do the job so well for microRNA, which results in a lot of false positives and a potential misdiagnosis. The third issue was that the buyer of these kits needed to reveal the sequence of the microRNA. So in early-stage microRNA-based discovery platforms, this is the kind of information that people would not be so willing to reveal. So we asked ourselves, shouldn't there be a better way of doing this? And we explored the various technologies that were available, and we came up with a method that enabled us to shrink the entire workflow into a single tube reaction. And we have published data to show that our method is able to achieve a significantly improved reliability for microRNA detection compared to what's existing. And the best part is, we don't need you to reveal your sequence. In our first phase, we aim to launch 2 basic products, the 1-tube kit, as well as our handheld fluorometer. So let me illustrate briefly how our product works. So let's say you have a microRNA from a cancer cell, as well as a healthy cell. All one needs to do is to mix these reagents with our proprietary dye that we provide with the tube, put it in our fluorometer, and the target microRNA would produce a fluorescence, whereas there would be no signal for the healthy cell. So we aim to enter the market through the RUO segment first, which is estimated at USD 160.5 billion, and eventually we want to go into the other diagnostics, followed by the cancer diagnostics market, which is valued at 1.2 billion and growing at a rate of 14.8%. So, the microRNA opportunity. Very recently, microRNA has been gaining a lot of traction. Early last year, Johnson & Johnson collaborated with 15 organizations, 4 of which were in oncology, and 1 being local startup Miraxis, who similarly employs a microRNA-based technology to develop solutions for lung cancer. And late last year, Miraxis and VentureCraft partnered together to form ARC, where they were launched with a 40 million Series A funding. So, what makes us different from our competitors? We are able to provide an integrated 1-tube setup for the reliable detection of microRNA through our 1-tube kit, as well as our fluorometer. We aim to target mainly research institutes, followed by public health, as well as biopharmaceutical companies, and we aim to reach them through distributor channels as well as clinical partnerships. So far, we have managed to conduct 20 to 30 interviews with these institutes, and we found very promising and encouraging feedback. For example, Professor Justin Chu from Duke NUS mentioned how we could consider the use of our diagnostic kits not only for cancer, but hand, foot, and mouth diseases as well. So in early 2019, we aim to set up our lab and continue developing our prototype, and we hope to get our product out by late 2020. Matien and I, we are a group of chemists, and our core technology lies in the development of sensors for the detection of biomolecules like DNA and microRNA. We are well supported by veterans from not only academia, but the industry as well, and we have clinical contacts to provide the network. So, microRNA, as you can see, is a huge and growing field with immense interest, and we want to raise the benchmark for early stage cancer diagnosis. But to do this, we need 1 million in funding to complete our product development, set up our innovation lab, and to conduct our clinical trials. Join us early, and let's eliminate late stage diagnosis. Thank you.
Any questions from the floor? Shikaresh?
Excellent presentation, very encouraging. So my question is specific to the organs. Is your technology applicable to specific organs, or it can be used all across? And I have a follow-up, but let me hear that first.
Okay, to answer your question, yes. As long as we have the microRNA sequence that's identified to have a strong correlation with that particular disease, that's by organ, do you mean organ, a specific cancer type? Yeah, so as long as
Not cancer type, location of the cancer. It can be lungs, it can be breast cancer, it can be all over the body, right?
Yeah.
So do they have a specific signature, microRNA? I'm not aware of that, so that's the question.
Yeah, so because these are studies that are new and being developed, a lot of microRNAs are being discovered and found to have a strong correlation. So as long as there's this specific microRNA for this type of cancer, we are able to reliably detect it. So we're enabling these people who are doing the microRNA discovery to do a reliable detection. So that's where we stand. That's where we position ourselves.
Okay, so I saw your the color of the thing changes if you have a cancer cell present.
Yes.
Now, that's something like an yes and no kind of a thing. Like, so what they do with that finding? What's the next step?
Okay, so it's not as simple as a yes and no. That was a simplified illustration. We are able to quantify the amount of microRNA. So in certain cases, different types of microRNAs are either upregulated or downregulated, depending on which type of cancer is present. And by being able to develop a panel that shows these changes, we're able to say with greater certainty whether a person is likely to get cancer or has cancer already.
Thank you.
Any more questions?
Yeah, so you mentioned you are quantifying the quantity of microRNAs in cancer versus healthy cells. So how big is the difference in terms of the quantity?
How big is the
like, is it a 2-fold higher or 3-fold higher, or how big is the difference in cancer versus healthy?
Actually, we are providing a microRNA detection kit, which could be used for the screening of the microRNA. So you mean the expression of the microRNA in cancer cell and the normal cell?
Yes.
It depends, because sometimes it's up expression and sometimes it's down expression. So we just regulate the expression level then to determine whether there is some cancer or not.
So the question is, how much is that? I mean, what's the scale of that up or down expression?
Okay, because different cancer types secrete different microRNAs, and different microRNAs are either up or down regulated, you cannot put a number to say that there is a certain percentage by which it is up or down regulated. It is specific to the microRNA and specific to the cancer type in question.
I'm asking this because Do you think in early stage cancer the difference will be big enough for you to detect?
It is because it has been shown to be significantly different, and a lot of papers have been published since 2002 to show that difference, that we have this technology today that we are developing. There's a lot of, you can actually check out the publication. It's been just increasing to show a lot of correlation between microRNAs and cancers and how they differ in healthy cells versus cancerous cells.
Maybe this question you can discuss in more detail at the booth. So any more questions?
I'm just asking the same question in other words. What is the sensitivity level of your detection in terms of quantity, like so many micrograms or femtomoles or something like that? Have you tested that?
Yeah, actually we are providing a reliable microRNA detection kit. So because the current microRNA kit based on the PCR could achieve 90% selectivity, but our method could achieve 99% selectivity. So
how different is it from the nanopore detection? Sorry, the device that we use for DNA or RNA measurement?
yeah,
Yeah, nanometer.
The nano drop, you mean? Okay, it's currently comparable in terms of sensitivities. We are able to achieve femtomolar detections.
Okay.
Yeah, to answer your question.
One last question from myself. You're using this primarily as a screening tool or a diagnostic tool?
now it's just a microRNA detection kit. But maybe in the future, after cooperating with hospital and pharma, we will develop some screening and diagnosis kit for cancer.
Thank you. Any more questions? Okay. All right. So let's thank our final team for the session. MIRNA, good job.